blockbuster research work from Team 2 called Development of Biofilm-Derived Biomaterials for Wound Healing and Tissue Regeneration. The bacteria used in the project is Klebsiella pneumoniae. Klebsiella pneumoniae has a road shape. It's gram-negative and usually has a capsule, but not always. The bacteria has some associated pathologies, such as pneumonia, urinary tract infections, bloodstream infections, wound or surgical site infection, meningitis, abscesses, and cellulitis. The main viral factors from the bacteria are the capsular polysaccharide, fimbrite, iron chelating compounds, siderophores, biofilm formation, and lipopolysaccharide.
And lipids, all derived from the bacteria themselves as well as from the host.
The process of biofilm has different steps. Let's see them. First, the formation starts with the attachment to the colonizing surface through structures such as surface adhesins as well as fimbriae and flagella. After this, the bacteria multiplies, generating microcolonies surrounded by cell-produced extracellular polymerase substance, or the acronym EPS, and continues with the production of the microcolonies, the biofilm maturation, and further organization. Lastly, we have the detachment of planktonic cells happening, giving the opportunity to them to spread and colonize or infect nearby or distant sites. Biofilms are regarded as a reservoir of bacteria during host infection due to their high level resistance to antibiotics, environmental stressors, and providing a reservoir for dissemination and further gene exchange associated with antimicrobial resistance. Okay, now let's see the related pathologies. In this case, we have very common urinary infections, the ones that are produced due to the formation of biofilms in the used catheters.
Wounds in Mexico are a public health problem that affects the quality of life of those who experience them. In recent years, the incidence of injuries in our country has increased. For example, according to data from the Ministry of Health, an average of 13,000 people are reported to have some type of burns each year. In Mexico, the direct monthly costs associated with outpatient wound treatment are around 46 million pesos, while in hospitalization they increase to values close to 2 billion pesos. The most recurring wounds are those that affect the skin, muscles, and subcutaneous tissue, with 26.6%, and diabetic foot ulcers, with 23.4%. Furthermore, another study revealed that the national prevalence of pressure injuries is 12.92%, which increases the risk of death by 4.5% in patients with comorbidities such as diabetes and hypertension. Epidemiological data in Mexico indicate that within a study of 357 patients from 14 primary care medical units, the prevalence is for traumatic wounds 59%, surgical dehiscences 12%, leg ulcers 11%, diabetic foot ulcers 10%, burns 4%, injuries due to pressure 3%, and anus and perineum wounds 1%. Regarding the anatomical location of the lesions, 66.2% were located in the lower extremities, 19.2% in the torso, 9.8% in the upper extremities, and 4.8% in the head and neck. So, what has been done to solve the issue? Multiple proposals have been implemented to treat tissue healing and regeneration problems, from the most traditional ones that involve the use of natural remedies such as honey or aloe vera, the use of modern tools such as gauze, bandages, plasters, or antiseptics, to much more current methods such as transparent compresses with specific properties depending on the type of wound, skin-friendly compresses with better adherence to the skin, or more sophisticated therapies such as negative pressure. Those more advanced treatments include therapy with growth factors, stem cell therapy, gene therapy, and biomaterials loaded with bioactive molecules that contribute to adequate healing. What hasn't worked to solve the issue? Rather than discussing methods that have completely failed to treat scarring, we can expose various disadvantages presented in the different proposals, such as the fact that gauze or plaster are not sterile and dry out the skin. Using temporary biological dermal substituents can transmit diseases. Immunological incompatibility, the limitation of not using if there is an infection, bleeding, or excessive sweating. The synthetic dermal substituents shouldn't be used if there is hypersensitivity to the product or wound infection.
The methodology of the proposal addresses as follows a study population that involves diabetic patients suffering from chronic ulcers, especially those with infected diabetic foot ulcers. The criteria are patients with diabetes mellitus type 1 or type 2, presence of non-healing chronic diabetic ulcers, signs of active infections, list of materials and reagents. We will need Klebsiella pneumoniae strain cultivate to produce biofilms, Triton soy broth and sodium chloride. Materials for biofilm extraction, centrifuge, sterile microtiter plate, shaker and 37 centigrade incubator. Reagents for extracellular polymeric substance purification, sterile distilled water, PBS, collagen, and growth factors. Clinical materials, sterile swabs for user sample collections, wound dressings, sterile scalpel, saline solution for wound cleaning. Analysis equipment, microscope, stereofactometer, autoclave for sterilizing materials, sonication equipment for EPS separation, and finally quantification kits like ultraviolet stain for biofilm analysis, cell viability, assay kits, and standard laboratory materials like pipettes, petri dishes, and vacuum tubes.
For the methodology, the objective is focused on developing a biofilm-derived biomaterial from Klebsiella pneumoniae, without bacterial cells, as a biodegradable and biocompatible scaffold for tissue regeneration in chronic diabetic ulcers. Phase 1. Cultivation of Klebsiella pneumoniae biofilms. Bacterial culture. Klebsiella pneumoniae ATCC 13883 will be cultured in tryptone soya broth, TSB, at 37 degrees Celsius and shake 120 revolutions per minute overnight. Biofilm collection. The bacterial suspension will be centrifuged at 5000 G for 10 minutes, and the supernatant discarded. The cells will be washed twice with sterile TSB, and the bacterial density will be adjusted to 1 million colony-forming units per milliliter. Biofilm production. 10 L of the diluted suspension will be deposited in wells of a sterile microtiter plate, incubated at 37 degrees Celsius for 24 to 48 hours to promote biofilm formation. Phase 2. Extraction and processing of the extracellular polymeric substance, EPS. The extraction. The biofilms formed will be processed using sonication and centrifugation techniques to separate the extracellular polymeric substance, EPS, from the bacterial cells. The absence of pathogenic bacteria will be ensured through sterile culture assays. Modification. The extracted EPS will be processed to create a biodegradable scaffold. Biocompatible modifications will be made, such as cross-linking with collagen and the inclusion of natural growth factors to enhance wound healing. Phase 3. Application of biofilm-derived scaffolds in diabetic ulcers. Experimental group. Diabetic patients with ulcers who will receive treatment with the biofilm-derived scaffold. Control group. Diabetic patients with ulcers who will receive standard treatment with dressings and traditional care. Clinical procedure. The biodegradable scaffold will be applied directly to the ulcers, covering the wound with biofilm-derived material and sterile dressings. Dressings will be changed every 48 hours, and wound healing will be monitored for 30 days. Phase 4. Analysis and follow-up. Healing monitoring. Daily measurements of ulcer size will be taken using digital images and image analysis software. Wound bacterial cultures will be conducted weekly to evaluate the presence or absence of infections. Residual biofilm quantification. Biofilm formation will be evaluated in samples of ulcer exudates using crystal violet staining and microscopy. This point is carried out to assess the efficacy of the treatment, that is, whether the biofilm formation was reduced and if there was an improvement in the healing of chronic diabetic ulcers. Final evaluation. Results will be compared between the experimental and control groups in terms of ulcer area reduction, presence of biofilms, and healing speed.